Sanibonani, Dumelani, Avusheni. Hello, Nda. Welcome guys to my class. Welcome guys to my class. Today's date is 22 March 2024 and then time here 33 minutes past 1. And then guys, thank you for all the viewers that I'm receiving and all the feedback, the WhatsApp group guys. Thank you. And then guys, as what we are pushing with our social media, it's a long walk to distinction. Long walk to distinction. Genetics and inheritance. Subtopic sex determination, sex linked inheritance. Today is Friday, as we have promised that per week we'll do this week, and then today we are here. So you can see that these topics, they are coming after concept of inheritance on the monohybrid crosses. So it will be much beneficial for you guys to know or to understand the concept of inheritance and the monohybrid crosses, because that is what we are going to use to do this sex determination and sex inheritance, or build up to our full topic here under CAPS topics. So okay, very information assessment. Then we have resources other than textbook to enhance learning, mind the gap, genetic crosses, past question papers, and then they have to revise cell structure and differentiate between chromatin and chromosomes, genes and alleles. Guys, that's what we have to know, requirement or requisite pre-knowledge. This is what you have to be using. So from there, our CAPS document here is sex chromosomes. We have to learn there's a difference between body chromosomes and sex chromosomes. Guys, by this time you understand, you did mitosis, you did somatic cells, you did meiosis, you did what do we call gametes and gametes. Yeah, you did gametes. So from there, let's move to our examination guideline. Everything is still pegged according to which color can we use here? Let's use purple. We love purple. Okay, and then sex determination, and then everything is still we are still moving order. From our monohybrid we are moving to sex determination. Why are we moving to sex determination after monohybrid? We learned that in monohybrid you have one trait that is being crossed with another trait. We used example about a color skin, a nose, and then where you have a co-dominant, intermediate dominant, or complete dominant. And then having a complete dominant as an example, we might say the heterozygous BB being crossed with heterozygous BB for the color black. And then there you know with monohybrid you can use a Punnett square, or you can use the crossing, and this crossing side, this strategy here, or you can use this strategy here to find out or to come up with the gametes, to come up with the final prediction, or if a baby, or if a father, or if a parent have a B, capital letter and small letter B, and the mother, she's having a capital letter B and small letter B, and then this one will meet here, capital letter B, capital letter B, capital letter B, small letter B, capital letter B, small letter B, small letter B, small letter B, and this one are the genotype of the F1. You know phenotype, and then you know monohybrid, and then from monohybrid, that's where we now use that knowledge to determine sex determination. We are just determining sex. And then from our examination guideline, they are saying that since you know that here that 22 pairs of chromosome in humans are autosomes, and then this we can find them on our somatic cells and our gametes. And then these 22 chromosomes in humans, they are called autosomes. It means that they are going to carry the characteristics, or they are going to carry all traits rather than the sex determination traits. or characteristics. So where now you have a 23 pairs, and then they said one pair of chromosomes are sex chromosomes or gonosomes. So it's either it's an autosomes, this one it's a 22 pair, and then or the two that are gonosomes or sex chromosomes. Then I was saying we have 23, and then in this 23, 1 up until 22, they are all going to be determining your skin color, your eye colors, your ear colors, your ear loops, your nose. Maybe you are going to have a big one or the small one, your mouth, everything is going to be determined from the chromosome number 1 up until chromosome number 22. And then at number chromosome 23, here we are talking about the karyotype, and you also have to know the karyotype and to determine it and to interpret it. And then at the chromosome number 23, that's where we are going to have the X and Y for a male, or X and X for a female. You know this, Amir. And then these chromosomes, this is what they are saying over here. And then males have X and Y chromosomes, and female have X and X chromosomes. Ne? These are sex chromosomes, and then they are determined by X and X. So what are we going to learn? What knowledge can you bring from the monohybrid and the heterozygous and the concept of inheritance that you can bring it here and use it? We can use the monohybrid cross to determine the sex of a baby, a sex of an F1 offspring. Like here we'll have P1, and then this will be genotype. Okay, and then this one, the phenotype will be male and then female here, and then they will cross, then they will cross, they will cross, and then they will cross, and then we will have XX, 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 XY, XY. Then the possibility is that 2 will be female, 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 and 2 will be male. So chances are 50% female, 50% male. You know this, Akir. Hence now you learn sex determination. Sex determination, we are using that information from monohybrid, bringing it down here to determine sex. Ne? And then with this info, with this sex determination, that's where we are going to link it with sex-linked inheritance, because now these chromosomes that we are sharing here, or this the sex that we are sharing here, we have a whole chromosome on it. We have chromosome like this. This one will be X, and then I like to do Y like this. I like to do Y like this. After the replication, they will be like this, ne? And then if they are like this, the X1, if they're like this, you see that this is the chromosome, and then in sex determination, we are just determining whether the X1 will meet with the another X, or the Y1 will meet with the other X, that side from the mother side. But if we look at these chromosomes, the top one, they also have the locus here, and then we know that in this locus we have a specific allele. In this specific allele we have a specific gene. So when these genes here that are found on our X chromosomes, this X chromosome, I will say X chromosome because Y doesn't contain those alleles, or those space, those locus, or those position to house those genes. So we'll say when these genes that will inherit with these X chromosomes, those genes, they are called sex-linked genes. Ne? They are called sex-linked gene. We call them sex-linked gene. So sex-linked inheritance, we say that this inheritance is the process, or is the action of inheriting genes. Ne? So it means here we are inheriting sex-linked genes. So these genes, we are going to find them only at the X of the female, of the male, or 2 X's of the male. Why? Here's the Y here, because the Y here doesn't have the space to code for that. You'll see when we do the karyotype and what. And then they are saying, let's move on. Let's cover our examination guidelines, and then we move with order. And then they will say differentiate between sex chromosome, which are gonosomes, and autosomes in a karyotypes of a human male and female. I talked about this, and we will have 1 2 3 4 5 6 chrome up until 23. When now you will have to determine the gonosome is the last pair, number 23, and the autosome is the chromosome 1 up until chromosome 22. Then you will learn. I will show you how to do this karyotype, how to interpret it. But when I bring the gonosomes and autosomes knowledge to our next explanation. Thank you. And then the last part under sex determination, we will have to represent, to do a representation of a genetic cross to show the inheritance of sex. Guys, the genetic cross, they are saying something like this. This is what I was representing. And they also want you to represent a genetic cross to show the inheritance of sex, where now you should be able to do this crossing. If maybe they are saying Lindiwe and Manmang, they are having it's a family of two. Like a family of two, you know, there's a female and a male, and that the lesbian ones, we can't include them because the reproductive system, it won't carry the baby. So female and a male, Lindiwe and Manmang, they are having a beautiful family and they're having 2 sons. So Jigi Lindiwe, she's also pregnant again. So when as a student, use a genetic cross to represent the possibilities of the next third baby to be a boy or a girl. Give percentage. No. You can repeat that if you didn't get it. So when you will come and say P1, and then you say a phenotype, and phenotype, and then you say male, and then you say female, and then if you can write male Lindy, a male Andiswa, female Lindy, and then you go do genotype, you'll say female, our male is going to be XY, and then our female, she's going to be XX. Lindy, she's pregnant, and so we're going to have meiosis. After meiosis, we'll have gametes, and then these gametes will be X, Y, X, X. And then when you choose, do you want to do a Punnett square or do you want to do lines? Either way, we will have fertilization, and then fertilization will happen if you are doing the crosses, you do the crosses. If you do a Punnett square, I want to give all of them a chance, and I don't want you to cram one, although this one takes time, but I want you to know all of them. Gametes, you write gametes, your gametes, and then you X, Y, and then you do X, X, and then this X and X, they will X and X, X and Y, X and Y, X and X, X and X, X and Y, X and Y. Then from there, you go and write a genotype, and then genotype, you'll write them X, they will be XX, XY. Man, you see, I also need space. Sorry guys, I thought I won't. Okay, and then you will come here, and write. Don't focus on anything, focus on what I'm writing, and you don't focus on this other information. And then you'll come and write genotype, and then you'll write XX, and then we'll have XY, XX, XY, XX, XY. And then you know that phenotype will have a 2 female and 2 male. So chances is 50%. They required you to give the chances of the third baby to be a boy or a girl. Now you can tell them then the chances is 50%. The baby or the child or the offspring, it can be a boy or can be a girl. Doesn't matter if when you had 3 sons in a row. Doesn't matter if you have 3 daughters in a row. The possibilities are still 50-50 when we deal with sex determination. Are we together? Thank you. Then from sex determination, we also going to link it with the sex-linked inheritance. About sex-linked alleles and sex-linked disorders. So we have alleles. Then this is what I was talking about, this side, and This is what I was talking about. We will have alleles here at a sex-linked alleles. These are the alleles that are linked to a sex chromosome. Ne? From a sex chromosome we get a sex-linked alleles. The question will come and say sex-linked what? Okay, this sex-linked, how do we write? We know that, okay, we know that when we write alleles like this, we are representing the all autosomes, ne? And then when we want to represent gonosomes, or we are representing a sex-linked allele, how do we represent that allele? Because we have X and Y multiplied by X and X. Because it's a sex-linked allele, this allele is linked to a sex chromosome. It means on top of this X we'll have A of whatever, and then A, and then A. And since I told you here that why that we don't involve it into an allele, because it doesn't have space to carry those allele, it means these allele that are linked to a sex chromosome, they will be linked to these A's but not to the Y here. Okay, example. And then we also have sex-linked disorder. When this disorder, it just means when everything is in order, everything is a mashup. When everything is now in disorder, it means that now there is an unclear way of doing things. The machine is not doing correctly. There is a disorder. I want to give you an explanation. I want to tell you that there is a difference between a disorder and a disease. Ne? A disorder, just a thing is not working well. A disease, it's you are affected by something. Then okay, from there we will do this. Now you know that the sex-linked alleles, they are linked like this, and then we have sex-linked disorder where we have X, Y, and then X and X now being involved with this X carrying this genes here, the hemophilia and the color blinding genes. Okay, but genetics problems involving the following sex-linked disorder. You see, sex-linked disorder. It means that when this sex-linked, when the alleles that are linked here, they are associated with these disorders here, you are going to suffer that disorder. For an example, H hemophilia. H hemophilia is represented by capital letter H and small letter h. And then color blindness, capital letter B, small letter b. And then here it means that on our male, we'll have two possibilities. We'll have this H having a capital letter H, and then another male here, there's a possibility of him to be like that. But when we come to females this side, we can have H, we can have H and a small letter h, or we can have small letter h and a small letter h. You learn this when we move on. So from here we are good, guys. From here we understand our sex determination. How are we determining our X? How are we determining our sex? And then how are these now we have sex-linked alleles on top of the sex chromosomes. And then when these alleles, we have disorders, we know that dominant chromosomes. Okay, let's move on. Let me not. Then hemophilia and color blindness. Not only these two, ne? Not only these two. I will show you others. Here's the karyotype. Karyotype. So they will say karyotype, but karyo, how you cram it, you cram it. You don't cram explanation. Why now you cram what? You cram the weights, how to write them. And then sex-linked inheritance. I wanted to say that genes located on a sex chromosomes are called sex-linked genes. I talked about it. And then it means that we'll have a chromosome. This is chromosome, and then karyotype, and a karyotype, you will see it like this. Let's see which one was that one. Which one? Here, chromosome number 23. as you see that this one, they are homologous, both of them, it means it's X and X. Ne? The short one will be X and Y, and they will be represented like something like this. Sure, Majita? Sure. Let's move on. And then we have sex-linked genes. Ne? Genes located. Sex-linked genes. These are the genes located on a sex chromosome. Ne? And then from there, Y is not affected, X affected. Whether you get X from your mother or you get it from your father. Now, if this and this match, obviously you'll be a girl. And then you understand something like this. Ne? And then, okay, it's fine. And then we also have we have sex-linked gene. This sex-linked gene, they are going to produce out, they are going to give us a sex-linked trait, ne, or characteristic, ne. And then these characteristics, we are going to say these are the traits. These are the traits controlled by gene located at a sex chromosome. This gene, they are going to control these specific traits. And these traits or these characteristics are traits that sex-linked traits. Example, hemophilia. Hemophilia is a condition or a disorder. Because it's a disorder, ne? Understand, it's not a disease, it's a disorder where your body or where your blood can clot. We cut you now, you are going to ooze blood, you are going to release blood non-stop. You are suffering from hemophilia, or you have a disorder. Something in your blood, it's not working. Hemophilia means blood, from hemoglobin, and philia to attract blood. So when you are suffering from hemophilia, we said you are hemophilic. You get me? And then another one is color blindness. Color blindness, it's a condition or it's a disorder where you can't see colors. You see, maybe like for example, if this one is blood, this one is to do with blood, and this one is to do with eye. We are going to do a human eye next week. So color blindness, somewhere in your eyes there is a cone, and those cones, they work as a photoreceptor. So they recept a color and they tell your brain that the color that you are seeing is yellow. So when you have this disorder, because in that particular place there is something that is not working properly, it means that when you see a red color, let's just for example, you see a red color, and then you say, and your brain tells you that red color is yellow, you have a disorder. You see a green color, and then your brain tells you that it's a yellow, you have a disorder, because your brain cannot tell that whatever that your eye is looking at, it's green or it's yellow. It's a disorder, also controlled by a sex-linked trait or sex-linked characteristics, being controlled by a sex-linked gene. And then another example, I prepared some examples for you. Here they are. Color blindness here. And then it's caused by a recessive allele. So it means that when now you know by this time that our X and X will I would like to start with male. And then we'll say your X and Y will cross with X and X. Then here we are saying we did a recessive and dominant. So because here we are doing a color, and then they are saying we use B to represent a color blind, it means B will be dominant and small letter b will be recessive. Hemophilia, capital letter H will be dominant and small letter h will be recessive, and this is called by a recessive allele. It means that you have to have two of these recessive alleles for you to be affected if you are a female. But if you are a male, because Y, it doesn't play that game. Why it's not there to determine whether you have hemophilia or you don't have hemophilia. The moment you inherit an X from your mother that is containing a recessive for a colorblind or for hemophilia, when as a boy child, you are going to suffer that disorder. And then we also have a Duchenne muscular dystrophy. And then this one is also called by recessive. This is what I did. I said all these three of them, they are caused by recessive. But when we come to the syndrome, Fragile X syndrome, Rett syndrome, they are controlled by a dominant. So when now you can cram, if you don't want to cram all these names, you can just cram. When a question comes, the question will start by saying, what it's a sex-linked disorder. When now you know something that is sex-linked, ionamela X is going to quell this thing of mine. And then maybe they say a Duchenne muscular dystrophy, it's a sex-linked disorder caused by a recessive allele. And then use D example. I'm giving an example. Use D as a capital letter as a recessive. This time they won't tell you that dominant and what. I can know your knowledge is increasing again. They will just say use letter D as a representative of a recessive. When you know that when we represent recessive with D, we are going to use small letter D. And where is this small letter D going to be carried at the X? Small letter D, capital letter D, the person is fine. Small letter X, D, the person is something. We have something here. Actually, we said a male will only need a recessive one again for this to be affected. And then if the female, we see that the female is having a dominant one, it means that he or she is not suffering from this thing, okay, this muscular dystrophy. Ne? But when it comes to female, this female here, she's not suffering. Ne? How about this female here? Capital letter D, X, small letter D. How about this one? And then how about this one? How about this lady here? This one, we are agreeing that this one, she's not suffering. She's normal, Anir. She's normal, this one, because this disease is called by the recessive one. And then she is having a dominant one. It means she's normal. And then what if she's having a small letter and a capital letter? Is she normal or is she affected? The answer is not affected. She is not affected. But no, we don't end here. She's not affected. She is a carrier. She's not affected. She is a carrier. She's carrying a small d. She's carrying a small letter d. And then there's a possibility if we are going to say this one is going to cross with a normal father. There's a possibility of this Y to match with this small letter d, and they are going to produce a affected boy. Life science. You see, hence we said she's a carrier. And then when we say she's affected, she's this. When we said a woman, a hemophilic female, we are going to represent that hemophilic female like this. So you see, they can just come with a statement and say a hemophilic female, you know that she's homozygous recessive. Or they said a heterozygous female, you know that she is H and small letter H. So like the judges. Okay, you got it, you got that, you got the disorders, and then let's move on, guys. We come to our textbook. We understand how our textbook is telling us how to place these words, and then we move on. We are done for the day. We are not dealing with color blindness and hemophilia only. You see, I managed to come with 5 of them. And these syndromes, I told you, the moment you see syndrome, dominant. What syndrome is a sex-linked dominant? Because they can trick you. They can say a Fragile X syndrome is a sex-linked disorder. A father having a Fragile X syndrome, he's mating with a female, being a carrier. Then when you will have to know if she's a carrier, it means that will she be a carrier here? She will be affected. We won't have a carrier here because the dominant one will be it's either she will be affected and suffering here. Because when we deal with dominant, it will be dominant over the other. But when we deal with recessive, that's where we see a process of someone being a carrier, or a condition of someone being a carrier. So our textbook, Itomaka sex determination, but the following problem represent a genetic cross which shows inheritance of sex. A couple has 3 sons. You see what I was talking about? A couple has 3, and then but it shows they give you a scenario nyana, or a story nyana, or something to rely on. They say it shows a genetic cross which shows inheritance of sex. It means we are only dealing with XY multiplied by XX. So but a couple has 3 sons and the woman is pregnant again. They say the example that I was using. A couple has 3 sons and the woman is pregnant again. Show by means of a genetic cross what the percentage chances, what are the percentage chances of the couple having a baby girl. By this time you know chances 50-50. Even if if your mother has to come and wake you up, and then your mother, she's asking you if tomorrow you are going to have a baby, what chances of that baby to be a boy or to be a girl? 50-50. There's no way how someone would look okay, now okay, sharp, let's do this, and I want to explain something. I remembered something. And then here's the phenotype. You do the phenotype, you do the F1, you do the P1, you do the F1, you get your right meiosis between genotype and gametes, fertilization between gametes and genotype, you get your other mark. 1 mark. And then another thing, you come and say phenotype male, and then phenotype female, and then genotype XY, you place them there, and then gametes, they will undergo meiosis, and then they produce these gametes. 50% of sperms carrying X, and then 50% of sperm carrying Y chromosome, and then here either egg, because you know that even meiosis, 3 eggs, they degenerate and only 1. So possibilities that whether any one of those 3, one of those 4, it's going to survive, that egg is going to carry X. And then from there we'll have gametes, the Punnett square, X and X meet, X and X, Y, X and X, X and X, this X and this Y here. And then genotype, you write X and X, X and Y, very good. And then the F1 again, and then you write 2XX2 is fine. And then by genotype female, 50%, and then male, and then genotype ratio 1. Then they said give a percentage chances, and the compulsory mark, did the person give the percentage here chance? Yes, the person is saying the female is a 50% and the male 50%, and she managed to show it. We gave her that compulsory mark of the thing. Then what am I? You see? So when you know now the things at the what we call monohybrid, you understand now that this is what it's happening with our thing here. And then here they said the genetic cross above shows the percentage chance of having a boy or a girl is 50%. Yes. And then in human, there are the 46 chromosome. You know this by now. You're seeing this by now. You are old enough. We can't be teaching you something today and then tomorrow you forgot, and you can go back to grade 10. Did you know that life science is taken as a 3 years module? you did this 3 years, you did it grade 10, grade 11, grade 12. So this one is your third year. You are graduating now. So at the third year, if you are still learning something today and by tomorrow you've forgotten that thing, you have to go back. So grade 10. Then okay, it's fine. So then of the 46 chromosome, 44 control the appearance, structure, functioning of the body. Do you understand? And then these are called autosomes. We talked about this, your appearance, your figure as a lady, you understand, as a man, your masculinity, your voice, your waves, your what. It's controlled by these 44 chromosomes. Then those are called autosomes, and then the remaining pair determine the sex of the individual, and they are called gonosomes. In a female, the gonosomes are 2 large X chromosomes, and in a male there is a 1 large X and a Y smaller chromosome. They said you have to know this by head. XY multiplied by XX. This one male, female. And then this one is the pair number 23, and they are called the gonosomes. The rest, they are called the autosomes. We're not at the Ari, let's continue. And then each species has its own unique number, shape, and size of chromosome. This is called karyotype. I talked about this. And then here's the karyotype here. There's the karyotype, and then examine the 2 karyotype below to see if you can identify which comes from a female and which from a male. And then let's go. Number 23. Are these homologous? Now you know homologous. We will explaining homologous now and again. Are these homologous? Yes. If it's yes, it means we have a female. Ne? Because she's X and X. And then let's karyotype 2. Are these homologous? No. Then it means we have male. And then because it X and Y. And the Y are now you can see it. It's the color what I did say. Hemophilia, color blindness. I don't involve as ngeni lapho thina. Singamadoda. Asingeni lapho. Na iningi nabafazi nasifaka izigundwane zenu. I'm not sure if you understand that. After meiosis, an egg will have 22 autosomes plus an X gonosome. A female will have 2 large X gonosomes, as in karyotype 1, X and X, while a male will have 1 X gonosome and one Y gonosome, as like in 2. And then female, male, remove male wire aluta continue. Males that have 2 types of sperm. What 2 types of sperm we find in a male? We have the one that contains 22 and X chromosome, and the other one that contains 22 autosomes and 1 Y chromosome. Depending on which sperm reaches the egg, there is a 50% chance of the zygote being male and a 50% chance of the zygote being female. So here we understand that chances are 2, and those chances or those variations are being brought by a male. So if now my boy, my guy, you are going to have a family tomorrow, and then in that family this is the problem that also our fathers are facing today, that in a family we are only having girls in that family, and the father will start now going to take stamp to come and have a male son in that family. And that thing you understand, and then as a life science student you understand that the problem is with me, a guy. In this process ya sex determination, where are they knowing something there? Sex disorder. In sex disorder we have to blame females, but sex determination we have ourselves to blame, my guy. When it means that your Y sperms, you are 50%, your Y sperms are not active compared to your Y sperms. Reasons maybe the mitochondria thing. Reasons maybe the tails. You might find that the Y ones, they produce small tail, and then the X one, they produce large tails, and then hence you find that the X one move faster and then fertilizes the egg. And you know, after fertilization, after the sperm enters the egg, everything shut down, and then the Y, when by the time they reach there, they found out that no, the fertilization have already occurred. So even if you move from one female and go and marry another female, 5 of them, if you have that power, if all of them, those females, they will have those daughter in your family. So guys, you can also educate batho ba baholo mo, and we are also educating one another. We won't find us tomorrow saying hey, because now with my wife we can't have son, let me go and have stembo. No, if you ufuna stembo ari yekelele ukuthi hey, I want a son. When you are the problem here in this scenario, sex determination. But when we do sex linked inheritance, that's where the females batsenang. If you want to blame your wife, blame ka this, blame ka hemophilia, blame ka color blindness. By that time when you have a son, Anir, if you have a son and then your son your son can't see, he got it from their mother. From that thing, you took this from your mother, man. And then the mothers, they have this one. You took this from your father. Although most of the bodily characteristics are carried out, most characteristics are carried out on the 22 pairs of autosomes, there are few characteristics carried out on the gonosomes only. That's, for example, the gene for a hair growing on an inside of the pinna, this yellow ear thing, and then it's carried on a Y chromosome. So only men will have these characteristics. Then if we have XY multiplied by XX, and then this characteristics is being carried in this Y, this is that shortness that you see there, or the bottom thing here, we can carry this along the thing. We will have the thing for sex production thing, testosterone, what, and then somehow we can have these characteristics here, and then all our sons will be affected with this, because for them to be sons, they have to inherit this Y. That in this Y they have the ears and they have the hair at the ears. Check it. If your father if your father is having the ears, the hair inside the pinna, inside the ears here, check the sons, they also have it. No way. More you can say asivanaka. More you can say this one no asivanaka. If you have a boy and that boy doesn't have the hair, you can say asivanaka. So you see how beautiful this life science is. You see how we get to enjoy this life science of ours. You can now explain things in scientifically manner. Okay, certain sex-linked genetic disorder are carried on the allele found in the X chromosomes only. 2 of these disorders are color blindness and hemophilia. We did this. And then a person is a color blind if unable to tell different colors apart. You see, it's not like you are color blindness. We are saying you can't see a color. You can see a color, but you cannot differentiate. Or this one is yellow, this one is orange. Or you cannot differentiate. This one is brown, this one is maroon. Most us males, this one also affect us males mostly, or most us males, we are more affected by this disorder because we only carry the X, the Y only from the mother. Hence you find that girls, they are good in color telling. Boys, some boys, they can comment there. Even though I'm writing with a color red, I'm using a red pen to write here. On some boys, they're just seeing maybe a light black, maybe, or brown, or maroon, or purple, or whatever color. So as male, we suffer most of this sex-linked that affect a sex X chromosome, because we inherit only one chromosome. So if a female, she's a carrier, we talked about this. If a female, she's a carrier, it means there's a possibility of the sons to be affected here. But for that girl, for a daughter to be affected, it will require my father to be affected also. Then if my father is affected and my mother, she's a carrier, we can have a sister that is color blind. Ne? But if my father he's fine and my mother she's a carrier, and then my father she's also fine. Example, my father can see the difference between yellow and green, and my mother can see a difference between yellow and green, but not as their son, I can't see that different. It means that from my mother she's a carrier. She's carrying the disease, but she's not suffering from it. Ne? I hope you understand. For an example, red-green color blindness is caused by an absence of a protein that makes up the red or green cones of photoreceptors. I talked about this in the retina of the eye, resulting in the person not being able to tell the difference between red and green. You place a red here, you place a green here, they see the same thing. I note this green. Not this one is line. Shoulder. You do something like this. They can't differentiate here. If when both of them they are red, for example, if both of them to you they are red, it means that the disorder or the thing that is not functioning is the green one. And then if to you both of them they are green, it means that where there is a dysfunction, it's at the red receptors. Your eyes are seeing things, but your brain can't tell you that these two colors are different. So hemophilia, we talked about hemophilia, the inability of the blood to clot due to the lack of a blood clotting factor in the if the person were to cut themselves, the wound will continue to bleed until the clotting factor is transfused in a hospital. We talked about this, Amir. Okay, it's fine. Hemophilia, Reukata, something like that is going to happen. But color blindness and hemophilia is caused by a recessive allele. We talked about this here. And then on the X chromosome, normally shown as small letter B for a color blindness and a small letter H for a hemophilia. By this time, they understand that when you see these small letters here, you see that this one is a recessive. Hence, they are not saying, and then the H capital letter, this one will be dominant. They are not even telling you that. And then as a result, men who have only one English here. As a result, men who have only one X chromosome. Textbook. Men who have only one chromosome have a greater risk of inheriting these characteristics. As a result, because due to men having only one chromosome, because there's no way that there will be a chance where a male have X and X. So it's because as a result of male having only one X chromosome, they have a greater risk of inheriting these disorders. A woman, on the other hand, have a much lower chance of inheriting two X chromosomes, which both carry the recessive allele for the disorder. If a woman inherits one X chromosome with a recessive allele for the disorder, she is called a carrier. We talked about this. And then here's the genotype and the phenotype. They are the sex-linked. They are the sex-linked disorder. Then H and H, homozygous, dominant, normal. Then here small letter carrier, and then small letter hemophilic female, and then capital letter X, that's what I was talking about, normal male, and then small letter A, small letter HY, and then hemophilic male. And then let's go to our color blindness, BB dominant. Since it's caused by the recessive, it means that this female, because she's an X and X, she's a normal vision. And then when something like this, she's a carrier. Then X and X, small letter, she's suffering. The capital letter X and Y, you see, you don't write anything on our Y here. Capital letter B, normal, and then small letter B, colorblind male. Happy. And then here's the thing that I was talking about. Do not add any letter to the Y chromosome, since the Y chromosome does not have an allele to counteract the recessive allele for hemophilia and colorblindness. And it's not only these two that we are going to learn. And then in an exam, you may be asked to do other sex-linked disorders other than hemophilia and colorblindness. Unless stated otherwise, follow the same format as in hemophilia and colorblindness. It means we'll hang capital letter or small letter, and here we'll hang capital letter or a small letter, unless stated otherwise. Then this will be your activity. You will do your activity blood. We'll do it next week. And then the activity, let me do number 3. Let's do number 1 and number 2. Number 1, Iri. Hemophilia, it's a sex linked. You see, whether you know what is hemophilia or don't know what is hemophilia, they will tell you whatever we are talking about now, it's a sex linked. You know that we are going to link them somewhere. We are going to link them on our sex chromosome. Disease. They say it's a disease, but now that's not disorder. There's a difference between disorder and the disease caused by the presence of a recessive allele, capital small letter H. Have a normal father. A normal father, it means that the person has a capital letter H and a heterozygous mother. You have the both. If you have children, have to construct a genetic cross to determine the possible genotype and phenotype, our compulsory mark of the children of the parents. Then what are we going to do? We are going to say X. You are going to start by P1 and P1, and then P1 phenotype, male and female. And then you're going to say male and female, and then X, and then gametes XY multiplied by XX. Don't forget to add the sex linked, and then here, because they're saying it's a normal, you have a capital letter H, and then this one, she's a heterozygous, capital letter H, small letter H, and then they will cross. You do the meiosis. They will do the gametes. After doing the gametes, fertilization. After fertilization, we are going to have X with capital letter and capital letter. Then we're going to write F1 generation, genotype. This will be the genotype. The second one is going to be X capital letter and X small letter. The third one is going to be X capital letter and Y. Then the fourth one is going to be X small letter and Y. I hope you see it. And then phenotype, we are going to have a normal. Here she's going to be normal, and then here she's going to be a carrier, hemophilia carrier, and then here he's going to be a normal boy, and then here we are going to have a hemophilic boy. Then we determine the possible phenotype and a genotype and phenotype of the children of the parents. These are the phenotype and these are the genotypes. You don't know what is happening? Go back and do the Punnett square. Understand what is happening with mono hybrid, and then understand what is happening with sex linked, and then sex determination. But explain why the chances of a man having a sex linked disorder, any disorder, explain why the chances of a man having a sex linked disorder is much higher than it is for a woman. That will understand that a woman have 2 X chromosome and a male have only 1 chromosome, 4 marks and then for a female, since most of these sex disorders are caused by a recessive alleles, it will need 2 X chromosomes with linked recessive disorder for a female to be affected. And then for a male, it will only need 1 X chromosome linked with the allele to be affected. You place it anyhow you place it, we see that you understand, we give you these 4 marks, guys. Don't say much, don't say a lot of things, but say much. Here's the example. Here's another example. When now you can't come here and be like, hey, because a mother, what, 50% ngwana, 50% son. So if 50% son, it means that the man is You bring this back to monohybrid. You bring this back to sex determination. You tell us that you understand the difference between a male and a female gonosomes. And you understand that when we deal with a sex-linked in disorder, females will need 2 X's to be affected, and male will only need 1 X to be affected. So chances there are more for a male to be affected compared to a female to be affected. So when you do number 3, and then after number 3 we'll come and do this together. If I don't have time tomorrow, I'll send the answers go WhatsApp group. Then I like this, I like 3.3, go and do it. 5 minutes left. Thank you guys for your time. Thank you guys for listening. Don't forget to like, guys. Don't forget to share to others, guys. Share, let's help one another, guys. We don't want to find things of where nakabo malas you say hey kikrile sipule, hey I got a bari, it's because I met this guy go YouTube, and let's all of us sit down and say we met, or when you met him, we will applaud you for bringing this information to other students. There's no thing now I got my corner, I will use my corner and use this information to outshine my other classmates. All of us, let's use it. Whether you are from where, use it. Download this, go into the school, download from the YouTube Wi-Fi school, go back home, watch it at home. Plug in your earphones, listen. Volume how fast and listen to Mr. Paper. I'll explain life science. Then let's run and go to our exercises. I also, guys, do some exercises so that where are we? And then but the diagram below represent the chromosomes from the human somatic cells of the 2 individuals who are twins. Individual 1 and individual 2, they are twins. Happy. What are the somatic cells? Somatic cell, by this time we know that somatic cell, they are the body cells, and then the other cells will be the gametes, which will be the sex cells. You get me? You have to explain that. When they ask you what is heterozygous, for you to explain what is heterozygous, you have to know what is homozygous. That's what they are saying. For you to explain what is somatic cells, you have to know that there's 2 types of cells. So number 2, without wasting any time, name the specific type of chromosomes numbered 1 up until 22. 1 up until 22, they are saying they have a specific name, but name the specific type of a chromosomes. There are autosomes. And the other 23 here, it will be the gonosomes. But they didn't ask you that, but you know it, and you're just flexing. And then number 3, each of the pairs shown is a homozygous pairs of chromosome. It means number 1 here we have 2 homologous chromosomes having homologous pairs during zygote formation. Was that a question? Each of the pairs shown is a homologous pair of chromosome. But state the origin of each chromosome in a homologous pair during zygote formation. Now we understand that zygote formation will get 23 and 23 chromosomes, and then this will match again. And then we state the origin. Tell us where are these 23 chromosomes coming from, and where is this 23 chromosome coming from? They are coming from the parents. And then you can say father, or you can say a male and a female. That's what they want. The origin, where the thing. And then number 4, explain one observable reason why the two individuals are not identical twins. Explain one observable. It means that this reason that you are going to explain here, everyone will see, everyone can see it. Even if you take this and you take these two individuals, this karyotype, and take them to your sister or to your younger sister and say, show me difference between these two, they can observe it. Where? Chromosome number 23. This is the one observable reason. Chromosome number 23, this one is X and Y, because this one is not homologous to the other pair. And then this one, individual number 2, it's X and X, because they are homologous. And why the two individuals are not identical twins. They are not identical twins between individual number 2, she's a female, and individual number 1, he is a male. Due to these reasons, and the chromosomes, they are not homologous here. Here they are homologous. Then X and X. 1 mark, 1 hour mark, we have reached it. And then that's it from me. Thank you guys, until we meet next time. Don't forget guys to share these videos. Let's share these videos guys. It will also give me spirit to say to continue with it. And thank you for those who watched. Thank you for subscribers. We are at number 3 subscribers guys. Last time I checked, we're at number 3. So guys, thank you for those 3 subscribers. We will celebrate our 10 milestone, our 50 milestone, our 100 milestone, our 1,000 and 1,000 milestone. Last question. Bari, color blindness is a genetic disorder caused by a recessive allele on a X chromosome. It means that we are going to have our XY multiplied by XX. It's a sex-linked anir. And then this color blind is caused by a recessive allele. Okay? And then Mari, a color, we don't write anything. This is what we can write. We are still going to link our allele here. And then number 1, but a color blind man marries a normal vision woman who does not carry the allele for the color blind. And then we know that for the color blind, unless stated otherwise, we will use B. Do a genetic cross to show the possible genotypes of their children. Use B for the normal vision and small letter b for the color blindness. And then by a color blind man, it means here is having a small b, marries a normal vision. It means it will be B and B. From this, you get your right, Anir. You successfully linked, and then mind you, our Y, we don't touch it. Then from there, you will write a P1. Let me waste this time. P1, we will write phenotype, and then you will write here a color blind male, and then normal vision female, or we write male or woman, and man or woman, but the proper way is to female and male. Okay, it's fine. And then there you'll come and say genotype, and then this will be a genotype here. And then you'll say meiosis, and then meiosis will have gametes. So should we do Punnett square or do crossings? Yeah, the crossings now. Which one can we do? And then from there you'll write fertilization. Fertilization, there's a possibility of this. We need, guys. Punnett square will do it next time. Now we are still doing our crossing Spider-Man thing. An egg and a sperm, and then this one will have X and X small letter B. You start with capital letter B, and rule of thumb here, it will bite us. Since it's this B and this B, we'll have X and X, capital letter B, small letter b. Then our second one, same thing, capital letter B, small letter b. The third one will be X, capital letter B, and Y. Then the last one will be X, B, capital letter Y. And then this will be genotype. Anir. This will be genotype. And then with genotype, you will say F1, and then you will say phenotype, and then phenotype, you will say this one, is she a carrier, is she colorblind, or is she a normal baby? No, she's a carrier for a color blind. And then this one, is she a carrier or she's a carrier for color blind. And then Majita, on the boy side, are they normal or are they affected? We have normal this side. We have normal, and we have normal vision. And then the question says, do a genetic cross to show the possible genotype of the children. And then here's the genotype, the compulsory, you get it. This one out of 6, you have 1, 2, 3. 1, 2, 3, 4. This one is not here. 1, 2, 3, 4. I can give you 4, 5 year phenotype, and 6, 1 year crossing. Then you get your 6 out of 6. So you see the compulsory one, where's the compulsory one? The genotype. This the genotype that we want to see. Ne? So from here, good night guys. Good night guys, for those who will be watching this during night. And then until we meet again, call me 071 190 6168. From me, people, have a lovely weekend. Have a lovely Friday. Until we meet again, we can meet Monday. Monday we'll be doing, let's do a final recap. Monday we'll be doing the dihybrid crosses and blood grouping. So there's a possibility that I will do a follow-up every time before I start my lesson. I'll start with a follow-up of a previous, and then we move to our next. And then here, if we don't meet up, if I don't record a video tomorrow, it means that we are going to meet come Monday, guys. So good night, have a lovely sleep. Bao nwang, guys. Those who are manifesting, and those who are now busy drinking at this moment, at this time, doing grade 12, ask those who are upgrading, ask those who came back for the second time. They will tell you it's very much painful to waste your time. Everyone is given the same weeks, everyone is given the same time. So we prepare this, we get this, we pass this, and then after that we enjoy. NFSAS is going to give you a stipend. You are going to be paid each and every month in university. Going to chow that money. Bafunza Rushaka, they are going to be given money, stipend, 8,000 every month. Chow it. So you can now relax now. For sure, at this moment, unless maybe you are pushing something somewhere, there were people that are trading and what, we can't look down on you guys. Unless you are doing that, 8,000. There's no way you are getting that unless your parents are giving you 8,000 every month. But there's a possibility that next year you'll be buying us a cool time here, saying, I got distinction. Come Mr. Paper today. Today I'm a doctor. Today I'm a dentist. Today I'm a what. Today I'm a opti. Mr. Paper, you can come without even having a medical aid. I mean, we don't even have a medical aid. You can come with or without medical aid. We will help you. It was of your helps that we are here today. So let me not waste your time, guys. Thank you for those who subscribed. Thank you for those who are viewing our videos up until the last minute. Like, guys, comment that we are done. 1 week down. How many to go? Let's count. 9 to go. 1 week down, 9 to go. Thank you, guys. Peace. I love you. Peace.